My name is Yijun Wang. In this presentation, I would like to share with you my research measuring the impact of pineapple juice on quantitative liver MRI metrics, which are iron-corrected T1, iron, and PDFF. This work was funded by my employer Perspective. The motivation behind this research is to look at this procedure called magnetic resonance cholangiopancreatography, MRCP. And this procedure mainly is to look at the biliary structure. And it's a highly T2 weighted sequence. And therefore, the fluid in the bile and the bile ducts and the gastrointestinal tract were all showed bright under this T2 weighted sequence. In order to increase the visibility of the biliary system, we can use negative contrast agent to suppress the signal from the gastrointestinal tract. But these contrast agents are expensive and not very palatable, so an alternative can be used. Pineapple juice, because it has a very high concentration of manganese, when patients take it through the gastrointestinal tract, these manganese coated through the lumen and therefore decrease the T2 signal for those structures and increase the visibility of the biliary system. So as you can see from the right-hand side, both the stomach and the associated artifact, which was introduced during the reconstruction procedure, were both hugely reduced after pineapple juice were given. However, for abdominal MRI, clinicians might also want to investigate liver functional metrics. But then the effect of pineapple juice is not widely studied for these liver metrics. So the main goal of this research is to see, does pineapple juice affect liver metrics? And if this is a yes, do we need to do something to correct for it? In total, there were 30 participants' data being analyzed. All of them underwent 2 scans with and without pineapple juice. These sequences allow us to retrieve metrics such as CT1, PDFF, and T2 star. We then use these values to conduct statistical analysis. So these are the CT1 and T2 star metric maps acquired at the slice level of the liver. Other than the stomach being filled with pineapple juice, we can't find visible difference in terms of color maps between pre-pineapple juice and post-pineapple juice. We can use bar plots to help us visualize the trends introduced by pineapple juice. The height of the bar represents the differences post-pineapple juice minus pre-pineapple juice. So for CT1, we can see for both field strengths, there is a slight increase after pineapple juice injection. CR1 was derived as the reciprocal of CT1 and therefore showed the opposite trend. For iron, only at the 1.5 Tesla data, the population mean showed a slight decrease, but this is not the case for 3T. For PDFF, both field strengths would found a slight decrease of the PDFF percentage after pineapple juice ingestion. We then conducted statistical paired test and found out that only CT1 and CR1 were found to be significantly different after pineapple juice ingestion, but this is not the case for PDFF and iron. In order to answer the second question, is the effect need to be corrected for? We collected data from a previous repeatability experiment and compared it against pineapple juice experiment with Bland-Altman Analysis. The only difference between 2 study designs is the introduction of pineapple juice. So Bland-Altman Analysis allows us to see whether the difference between 2 scans are actually due to random. So we compare the limits of agreement between the pineapple juice and repeatability experiment, and we found that the variation found in pineapple juice experiment is actually smaller than the measurement variation acquired through the repeatability experiment. So in other words, there's no need to correct for the effect found in pineapple juice experiment. If we have a detailed look at the metric maps for the diseased participants in our study population, there's also no visible difference between pre-pineapple juice and post-pineapple juice. This is sharing the same pattern as the healthy volunteer, which was shown on slide 4. In conclusion, the findings from this study can be applied to a full dynamic range of participants' health status. Thank you very much for your attention. If there is any further question, please feel free to reach out to me through email or any social channel.